Welcome back to the deep dive. If you've spent any time researching health and fitness lately,
Yeah.
you have definitely been bombarded by the collagen trend.
Oh, yeah.
It is absolutely everywhere, promised as, the solution for glowing skin, pain-free joints, faster recovery, all that stuff.
Mm-hmm.
But if you're an evidence-minded person, you've probably noticed a real problem. The scientific evidence is, well, it's legitimately messy.
It really is.
It's leading even highly reliable experts to disagree pretty fundamentally on its value.
It's that classic information paradox, isn't it? On one side, you have these, headline-grabbing meta-analyses suggesting massive benefits.
Right. Big claims.
But then on the other side, you have critical scientists who are questioning the methodology, the funding, and, crucially, the applicability of those results.
Yeah.
So our mission today isn't really to give you a simple yes or no answer. It's more to arm you with the critical thinking skills to evaluate the data yourself.
Exactly. We're zeroing in on two major areas here by reviewing two very recent high-profile systematic reviews. One's focused on cosmetic outcomes for skin,
Right.
and the other is focused on athletic and lifting outcomes. But before we get into the weeds on those, let's just quickly establish what we're actually talking about.
Good idea.
Collagen isn't just some boutique supplement. It's actually the most abundant protein in the human body.
Absolutely. It makes up somewhere between, 25 and 35% of your total protein content.
Wow.
Yeah, it's the foundational scaffolding, provides structural support, rigidity, tensile strength to all your connective tissues, tendons, ligaments, bones, skin.
You dirt and stuff.
Pretty much. We mainly talk about types I and 2 and 3 because they're the most relevant for musculoskeletal tissue remodeling.
Okay, so what makes this particular protein unique, and why do supplement companies claim we need, more of it?
Well, it really comes down to its amino acid profile. Unlike, say, whey protein, collagen is disproportionately rich in just 3 amino acids. Glycine, proline, and hydroxyproline. But that concentration also means it's incomplete. It lacks tryptophan altogether, and it's very low in critical essential amino acids like leucine, isoleucine, and valine.
Ah, the BCAAs.
Exactly, the ones your body uses to really stimulate muscle growth effectively. Collagen is just not great for that specific job.
Got it. And we really can't discuss collagen structure without hitting its sort of non-negotiable chemical partner, vitamin C. The sources really emphasize that connection.
Oh, this is absolutely vital. It's basic biochemistry. Vitamin C is essential as a cofactor for the hydroxylation step, adding that OH group to proline and lysine.
Okay.
Think of vitamin C like the chemical glue that lets those building blocks lock into place and form that signature stable triple helix structure.
Right, the shape is key.
It is. Without that specific glue, the structure destabilizes. And that's precisely what leads to the catastrophic tissue breakdown. Bleeding gums, tissue disintegration that defined scurvy historically.
Makes sense. So if you are taking collagen, maybe just a modest intake of vitamin C, say 50 milligrams or so, should probably be taken alongside it, just to ensure the body can actually use those building blocks.
It's like a cheap insurance policy, yeah. It supports the optimal synthesis of the material you're trying to build. Makes sense to include it.
Okay, so that leads us right into the central debate. Why supplement with collagen peptides specifically? If it's an incomplete protein and our GI tract is designed to break down long protein strands anyway, what actually gets absorbed?
Right, that's the key question. Your body definitely does not absorb long strands of intact collagen. That's just not how digestion works.
Okay.
It gets broken down primarily into individual amino acids, like any other protein source. However, and this is the caveat that drives the entire supplement industry.
Ah, here it comes.
Yeah. There's some evidence showing that very small chains of amino acids, specifically dipeptides, that's 2 amino acids, and tripeptides, 3 amino acids, like the specific one, glycine, proline, hydroxyproline.
Glyprohip, right.
Exactly. Those might survive digestion and actually make it into the bloodstream intact.
And this is where the theory of the unique signal comes in, isn't it? The main rationale for paying extra for collagen peptides instead of just, say, buying a cheap bag of glycine powder.
Precisely. The hypothesis is that these specific intact peptides act as a unique signaling molecule. They somehow alert the connective tissue cells, the fibroblasts, to ramp up production of new collagen. The idea is they send a message in a way that free amino acids alone wouldn't. It's kind of analogous to how leucine sends a strong signal for muscle protein synthesis.
Okay, I see the theory. But if that signaling is the key differentiator, the data challenging it seems pretty significant. The sources we looked at point to some tightly controlled human trials that look specifically at this question.
They did, and honestly, the results kind of dampened the initial optimism. When collagen supplementation was compared head-to-head against an equivalent mixture of just the free amino acids.
Okay, a direct comparison.
Yes. Neither supplement showed a clear advantage in promoting the actual synthesis of muscle protein or connective tissue protein.
Really?
Yeah. In those specific studies, that supposed unique signaling from the intact peptides didn't translate into a detectable functional advantage for synthesis rates.
So essentially, we might be paying a premium for a signaling effect that the data, at least so far, can't definitively confirm exists in humans in a meaningful way.
That's a fair summary of that specific line of evidence.
That really changes the value proposition, doesn't it?
Yeah.
If the unique signaling is questionable, why does the argument for collagen persist so strongly? Is there another angle?
Well, it often comes down to dietary context. The argument goes that we eat vastly different foods now than we did historically.
How so?
Traditional diets included far more collagen naturally through things like broths, organ meats, eating the whole animal, bone-in cuts. Modern Western diets tend to prioritize lean muscle cuts.
Right. Chicken breasts, lean steaks.
Exactly, which means we consume much less of the specific amino acids that make up connective tissue.
You mentioned glycine earlier. Is that considered the biggest gap in modern diets?
It often appears to be, yeah. Glycine is the most abundant amino acid in collagen, and it might actually be conditionally essential, meaning the body's own synthesis processes often can't quite keep up with the high demands of turnover, especially under certain conditions. Some estimates suggest the requirement just to support collagen turnover is quite high, perhaps more than 10 grams daily.
Wow, 10 grams is a lot.
It is. So if you're not regularly consuming things like bone broth or gelatin, supplementation advocates argue that collagen peptides help fill that specific high-turnover amino acid gap, particularly for glycine.
Okay, that makes sense from a building block perspective, even if the signaling part is murky. Let's tackle the evidence itself now, starting with skin. This is the cosmetic deep dive.
Right.
We looked at the Munga Park meta-analysis.
Right.
That pooled data from 23 randomized controlled trials looking at hydration, elasticity, and wrinkles, typically over about 12 weeks, usually using around a 3-gram daily dose.
And if you only read the abstract of that meta-analysis, you'd be sold immediately.
Oh, yeah.
The initial pooled analysis showed really substantial benefits across the board. For elasticity, for instance, they reported a highly significant standardized mean difference, an SMD of 1.02.
Okay, and for context, an SMD of 1.02 is?
That is a massive effect size in this kind of research. It suggests a huge, almost undeniable benefit based on that initial pooling.
Okay, wait. That sounds almost too good to be true for just a supplement over 12 weeks. And this is where it gets really interesting, because the researchers didn't stop there. They applied a critical piece of methodology called a sensitivity analysis.
Yes, and this is the moment the whole picture flipped. A sensitivity analysis basically means they systematically removed the outlier studies, the ones showing extreme, sometimes maybe unbelievable benefits, to see if the overall conclusion still holds up without them.
And what happened?
When they did that, those huge positive results became much more modest, or in the case of wrinkle reduction, actually became statistically non-significant altogether.
Wow. So they essentially removed the home run studies, the outliers, and suddenly the overall picture collapses into meh.
Pretty much.
That is the definition of a skeptical finding right there. And they found huge variability between the studies, too.
They did. We have to talk about heterogeneity, which is measured by the statistic called I-square, 1222.
Right.
This tells us how much the results of the different individual studies vary from each other. For elasticity and hydration, the pi-2 values were over 90%.
Okay, for those of us who haven't run a meta-analysis lately, what does an I-2 over 90% really tell us about trusting that pooled average result?
It means the results are so variable. You have some studies showing a massive effect, others showing basically none, that pooling them all together is statistically problematic. It's like combining apples and oranges and then claiming, the average fruit flavor.
Gotcha.
When you have that much heterogeneity, the average result is inherently shaky and probably isn't reliably applicable to the general population. The underlying studies are just too different.
Okay, and then the real nail in the coffin here seems to come from the funding and quality bias assessment. This is where we potentially learn why the data was so varied.
Yeah, this part reads like a classic trope in supplement research, unfortunately. The critical takeaway here is that the non-funded studies, meaning the ones run independently of any supplement companies, reported non-significant effects on every single outcome measured. Hydration, elasticity, wrinkles, nothing significant.
Ah, the classic story. I feel like we need a little bell we can ring every time the study funded by the company selling the product finds it works wonders.
Right.
And the independent one? Well, not so much.
It's pretty much that direct in this case. In stark contrast, the industry-funded studies demonstrated significant improvements across the board.
Huh.
And similarly, when the researchers graded the methodological quality of the studies, the high-quality studies revealed no significant effects, while it was the low-quality studies that consistently showed those significant improvements.
That pattern is, well, it's incredibly telling, isn't it?
Yeah.
Funding bias and low quality correlating with positive results.
It raises major red flags about the reliability of those positive findings.
And frankly, even from a mechanistic perspective, I find myself deeply skeptical of these big skin claims anyway. The dermis, the layer where the collagen actually needs to be remodeled for wrinkle reduction and elasticity, it turns over extremely slowly.
Very slowly.
The idea that just 12 weeks of taking a few grams of peptides can substantially reverse years of aging damage or sun damage seems biologically, well, implausible.
It certainly clashes with our understanding of basic skin biology and turnover rates. So for the listener who's primarily focused on skin health, this data strongly suggests that your time and money are much better spent on the high-impact alternatives first.
Like what?
Things we know work. Consistent sun protection, minimizing smoking and alcohol, getting consistent sleep, managing stress, and ensuring you have adequate total protein intake and micronutrients like vitamin C.
The fundamentals.
Exactly. Collagen supplementation for skin is, at best, maybe a marginal gain after you've meticulously locked down all those fundamentals. It shouldn't be the first thing you reach for.
Okay, that's a clear takeaway for skin. Let's pivot now to the fitness crowd. The lifters, the trainers, people interested in muscle and tendon health. We move to the Bischoff et al. Meta-analysis.
Right. This one looked at 19 trials combining collagen peptides with physical training. The dose was typically higher here, usually around 15 grams a day. Again, for about 12 weeks on average.
And what did this one find initially?
Again, the surface-level findings look statistically promising.
Yeah.
The analysis reported statistically significant improvements favoring the collagen group for things like fat-free mass,
Ooh, interesting.
maximal strength gains, and even some improvements in tendon morphology, although the certainty on that tendon finding was rated very low.
Okay.
They also noted potentially better reactive strength recovery 48 hours after strenuous exercise.
Right, if you're a lifter hearing increased fat-free mass and maximal strength, you're probably reaching for your wallet already.
Understandably.
Yeah.
But this is where we absolutely have to introduce the major caveat, what we often call the comparator problem.
Okay, what's the issue here?
Virtually all of these positive studies compared the collagen supplement group to a non-protein placebo group.
Meaning they got
Things like cornstarch, maltodextrin, basically just some carbohydrate powder with no protein content.
Okay, why does that distinction matter so fundamentally for interpreting these results?
Because collagen, despite its limitations as an incomplete protein, is still a protein. It provides amino acids.
Right.
If you are resistance training, which is a stimulus to build protein-rich tissues like muscle, and you're trying to adapt, providing any protein source, even an incomplete one like collagen, is naturally going to be more advantageous for building those tissues compared to providing zero protein, just a carbohydrate placebo.
Okay, so the comparison isn't really fair if you're asking about practical choices.
Exactly. The study isn't answering the question you, the consumer, likely need answered, which is this collagen powder better than or even just as good as my regular, cheaper whey protein isolate for supporting my training goals? It's comparing protein to no protein.
And presumably, when studies do make that more relevant, direct comparison against a high-quality, complete protein source, the supposed superiority of collagen vanishes entirely.
That's precisely what the evidence shows. The sources cite a key study that directly compared 35 grams of whey protein to 35 grams of leucine-enriched collagen peptides during a resistance training program.
Same protein dose, one complete, one enhanced, incomplete.
Yes, and the results were clear. The whey protein group showed a clear superior advantage for increasing muscle thickness. Collagen's poor amino acid profile, particularly the low leucine content, simply makes it a suboptimal choice for maximizing muscle protein synthesis compared to complete proteins like whey.
So for muscle building, it seems pretty clear-cut. Stick with high-quality, complete proteins. What about for connective tissue repair, like tendons and ligaments? That's often a big selling point, too.
Right. The verdict there seems to be that training itself, consistent, appropriate loading is the dominant stimulus for tendon adaptation. That's number one. Then, adequate total dietary protein intake matters, providing the general pool of amino acids. And ensuring you have sufficient vitamin C and iron, those necessary cofactors for the remodeling process, is also paramount.
So where does supplemental collagen fit into that picture for tendons?
It's really a tertiary consideration at best. It might provide some additional specific building blocks, maybe helpful if your background diet is low in those, but it should never be treated as the primary lever for adaptation. Focus on training, total protein, and key micronutrients first.
Okay, let's try to synthesize all this data for the listener. We've basically established two huge overarching reasons for skepticism across both domains, skin and fitness.
Yes.
For skin, the positive data seems heavily correlated with, well, industry funding and lower study quality. Take those away and the effect shrinks or vanishes.
Correct. That's the bias issue.
And then for lifting and fitness outcomes, the positive data largely relies on comparing collagen protein to a non-protein placebo, like cornstarch.
Right. The comparator issue.
Which isn't a very useful comparison for someone already consuming adequate protein.
Exactly. That is the intellectually honest summary of the current state of the evidence, I think. We have to look past just the statistically significant p-values reported in abstracts and really question the real-world applicability and the methodological rigor.
If you substitute the comparator, if you swap that cornstarch placebo for, say, whey protein, the positive results for muscle growth just disappear. You have to stay rooted in biological plausibility and relevant comparisons.
Precisely.
So, given that glycine seems to be the most likely rate-limiting amino acid for collagen synthesis, especially on a high-protein diet, and we know we need fairly large amounts of it for turnover, what about just supplementing with isolated glycine powder? Could that be a cheaper, maybe theoretically sounder alternative?
You know, mechanistically, that's a very attractive argument. Glycine is needed in large quantities for collagen turnover. Isolated glycine powder is cheaper than collagen peptides. It's more soluble in water, and it's generally easier to consume in those potentially necessary 10-plus gram quantities without GI issues.
Right.
So from a purely theoretical, let's supply the most needed building blocks perspective, focusing on glycine makes a lot of sense.
I sense a but coming. We need to maintain transparency here. This is still moving into the realm of educated speculation, isn't it?
Absolutely. We have to be really explicit here. This idea is currently speculative. To the best of our current knowledge, there hasn't yet been a high-quality study that's done a definitive, clean, head-to-head comparison between collagen peptides and an equivalent dose of isolated glycine supplementation, specifically looking at tangible connective tissue outcomes like tendon repair rates or actual remodeling in humans.
So we just don't have that direct comparison data yet.
We simply don't know for sure which approach, if either, is superior for those specific connective tissue goals. It's a gap in the research.
It's fascinating, though, that we can review the same body of data, these meta-analyses, and depending on how critically we assess the methodology and context, arrive at such profound skepticism. The core conflict really seems to be balancing what the studies technically show, yes, statistical significance in a controlled lab environment, versus what those findings actually mean for a real person in the real world, trying to prioritize their limited time, money, and effort.
That really is the crux of it. If your budget is tight and you're choosing between buying collagen peptides or, say, investing in better sleep habits or maybe fixing a potential vitamin C deficiency, the choice based on robust evidence seems pretty obvious.
Right.
We're questioning the applicability and relevance of many of these positive findings, not necessarily disputing the simple statistical calculation that, yes, 15 grams of protein is technically better for building protein tissues than 0 grams of protein from cornstarch. That part's trivially true.
So wrapping this up, what does all this mean for you, the listener? Maybe standing in the supplement aisle right now, looking at a wall of collagen products.
I think it means you absolutely must acknowledge the uncertainty here. It is genuinely possible to find studies and make an evidence-based justification both for and against using collagen, depending on which studies you cherry-pick and how critically you read them.
Yeah.
The most justifiable decision, in my view, requires intense scrutiny of the research context. Ask yourself, who funded this study? What was the comparison group? If the positive result seems to rely heavily on industry funding or a comparison to something like cornstarch, that positive finding should probably be discounted as functionally irrelevant to your practical goals.
Focus on the fundamentals first.
Absolutely. Nail the basics before chasing marginal uncertain gains.
That is the perfect actionable summary. And maybe here's a final provocative thought for you, the listener, to chew on as you go. If we know that getting adequate total dietary protein, applying consistent and progressive training stimulus, and managing high-impact lifestyle factors like sun protection, sleep, and stress already cover most of the body's needs for tissue repair and health, what specific non-clinical condition, so outside of maybe severe deficiency or a highly specialized wound healing scenario, would truly make supplementing with an isolated, incomplete protein like collagen the single optimal choice over just ensuring those fundamentals are covered?
That is the high bar, isn't it? That's the question that the next generation of cleaner, non-industry-funded, well-designed research really needs to answer clearly.
Yeah.
Until then, stick to the basics that science overwhelmingly supports.
Sound advice. We'll catch you next time for the next deep dive.